We now have our full-length DNA from our patent, so we're going to copy and paste that into two tools. One to optimize the codon usage for E. coli. So if we remember, this particular polymerase comes from Bacillus styrothermophilus, and the codon usage is likely to look reasonably different between the two organisms. So in order to optimize expression, we want to make sure that the use of codons is aligned to E. coli's kind of general usage, and this is important to make sure that there's an appropriate pool of transfer RNAs for the correct codons to bring the amino acids during translation and hopefully to speed up expression. Of course, sometimes that's not always wanted, particularly for things like polymerases, which can be insoluble when they're expressed. So they might need some time to fold correctly in order to maintain solubility, but we're going to control the speed of expression using environmental factors such as temperature. So we could incubate at lower temperatures if we find that's a problem, or we can use kind of more reliable and reproducible methods such as promoter sequences and regulatory elements, rather than relying on the codon optimization to do that piece. So the tool that we're going to use for the codon optimization is published here, Gene Design 3.0 from 2010. And here is the web interface for that. So we're going to go initially to codon juggling. So it's super simple to use. Paste your sequence here if we had the sequence properly copied. Select your organism, in our case it's E. coli, and then in our next step we'll see the results. So as you can see, there's several algorithms in use here. We are going to use the optimized version of the sequence, but if you scroll down, there's a less optimized version. There's also a most different sequence, which has only 60% identity to what we originally submitted, and a completely random codon allocation there. So we're going to copy the optimized one into Benchling and just into a new file. Which I've already done here. So one addition that we've made is to add an ATG start codon to the beginning, which wasn't included in the original DNA sequence in the patent. Okay, so that's our codon optimized version. The next thing that we need to do is remove restriction enzyme binding sites. We're going to use a tool published on bioRxiv in 2017, refactoring gene sequences for broad assembly standards compatibility. And so for our project purposes, we're using loop assembly, which uses only 2 restriction enzymes. It's one of the reasons that we chose it, also because it's an entirely open source DNA assembly method, and the plasmids required, the vectors, are available under the Open Material Transfer Agreement, which is really important for our project, creating an open stack for enzyme manufacturing. Those 2 restriction enzymes for loop assembly are BSA1 and SAP1. However, there are a whole bunch of other assembly standards, and so it would be desirable if our sequences were as compatible as possible with those different options, to give people the maximal choice over how they choose to use the DNA parts that we're creating. And so this particular piece of software takes all of the restriction enzymes used in the BioBrick, BGI Brick, MoClo and Golden Braid assemblies, plus some additional sites that you might need to move, like Chi sites, which can be recombination hotspots, and basically destroys all of them with silent mutations. And there is an online server version in the MIT web space, which you can see here. And so we're going to come back to Benchling and take our full-length optimized DNA. So copy that to the clipboard, move across to the synthetic biology gene standardizer tool, and just copy and paste in here, which is what I did earlier, but I'll just repeat. Organism at the top, we can choose E. coli. One sort of restriction of this particular tool is that at the moment it only includes E. coli or Bacillus subtilis. The code is available on GitHub, so if you wanted to add other organisms, that would be possible. It's all under an open source license. We're going to standardize for everything, bearing in mind that we're going to have to do a manual check for SAP1 because that isn't included in any of these standards at the moment, but BSA1 fortunately is. So we're covered there. Okay, so we're going to submit that sequence. So that's our refactored sequence, and if we scroll down, we can see the mutations that have been made to remove all of these sites here. For example, there were 1, 2, 3, 4, 5, 6 APO1 sites, and the A at position 2205 has been changed to a G. And so we could record that information. We're actually going to rely on the fact that we have both the sequences and we can compare at a later date if we need to. So we're going to copy and paste this entire sequence here, and then go back to Benchling and create a new sequence, which I did earlier, but I'm just going to add this one. And there we have it. So we can create a translation again if we want to see the corresponding amino acids. And if we scroll down, we can see that there has in fact been a SAP1 site introduced here. So we're going to have to change that in a second, but we're just going to keep scrolling down just to do a quick manual check on anything that has been left in. There's 2 SAP1 sites there. Okay, so we need to remove 2 SAP1 sites. Okay, so the SAP1 site is GAA GAGC, so we want to replace this with something that is not that, but still codes for E. So we can look for the amino acid codon table. Okay, so glutamic acid, GAA and GAG. So if we come back to our sequence, so we could change this to GAG. Which removes us at one site. We also need to do that again further down. So no change to the amino acids, but we've removed the SAP1 site, and on this occasion a couple of other restriction sites as well. Great, so as far as I can tell, that is all of our necessary restriction sites removed. There are obviously quite a few, so always worth a quick final check. Great, and now just to make absolutely sure, we are going to create a protein sequence. From this DNA, and we're going to create an alignment to our original. just as a final confirmation that neither our codon optimization nor our restriction enzyme removal have resulted in a different set of amino acids. Okay, so 876 identical sites, pairwise identity is 100%, so we're all good at the translated level, and we now have a DNA sequence that is codon optimized for expression in E. coli and also has any problematic restriction sites that might interfere with standard DNA assembly techniques removed.